there, I was just making a macromalad, or in other words, a macromolecule salad. What's a macromolecule? Macromolecules refer to the four major classes of biomolecules: nucleic acids, proteins, carbohydrates, lipids. In biology, macromolecules are regarded as large biological polymers. Polymers are made up of multiple monomers, which are individual molecules. Macromolecules are organic molecules, otherwise known as molecules in living systems, usually composed of carbon atoms in long chains or rings, to which are attached to other elements such as hydrogen, oxygen, or nitrogen. The first and most essential part to my macromalad is the spinach, a carbohydrate. Carbohydrates are the sugars, starches, and fibers found in fruits, grains, vegetables, and milk products. Carbohydrates provide fuel for the central nervous system and energy for working muscles. A monomer of a carbohydrate is called a monosaccharide. When two monomers are linked together, we call them a disaccharide. When three or more monomers are linked together, we call them a polysaccharide. The bonds between monomers when forming di and polysaccharides are called glycosidic linkages. Glycosidic linkages are formed by condensation reactions. Condensation reactions are when one water molecule is removed, in this case during the formation of the glycoside. Dehydration is a subset of condensation reactions. Next we'll add the protein, turkey. A protein is a complex molecule that does most of the work in cells and is required for the structure, function, and regulation of the body tissues and organs. Protein is predominantly sourced from animal products, but alternatives such as beans and nuts are a good source of protein as well. A monomer of a protein is called an amino acid. A protein has four levels to its structure: primary, secondary, tertiary, and quaternary. The primary level is the simplest level of structure. It's just the amino acids in a polypeptide chain. Amino acids are simple organic compounds that contain a carboxyl group and an amino group. Polypeptides are linear organic polymers consisting of large numbers of amino acid residues bonded together by peptide bonds in a chain, forming part of, or the whole of, a protein molecule. In a peptide, the carboxyl group, COOH, of one amino acid bonds with the amino group, NH2, of another, forming the sequence CONH and releasing water, H2O. The next level, the secondary structure, refers to local folded structures that form within a polypeptide due to interactions between atoms of the backbone. The most common types of secondary structures are the alpha helix and the beta pleated sheet. Both structures are held in shape by hydrogen bonds, which form between the carbonyl O of one amino acid and the amino H of another. A hydrogen bond is simply a weak bond between two molecules resulting from an electrostatic attraction between a proton in one molecule and an electronegative atom in the other. The tertiary structure is the overall three-dimensional structure of a polypeptide, and the quaternary structure is when proteins that are made up of multiple polypeptide chains, said chains, come together. Now it's time to add the nucleic acid-rich peanuts. Nucleic acids are molecules that allow organisms to transfer genetic information from one generation to the next. These macromolecules store the genetic information that determines traits and makes protein synthesis possible. A monomer of a nucleic acid is called a nucleotide. A nucleotide is composed of three major components: a nitrogenous base, a 5-carbon sugar, and a phosphate group. When nucleotides are held together by covalent bonds, they create deoxyribonucleic acid, DNA, or ribonucleic acid, RNA. In DNA and RNA, the phosphodiester bond is a linkage between the 3-carbon atom of one sugar molecule and the 5-carbon atom of another, deoxyribose in DNA and ribose in RNA. DNA is double-strand and is composed of 4 different nitrogenous bases: adenine, thymine, cytosine, guanine. RNA is single-strand and is composed of the 4 major nitrogenous bases: adenine, cytosine, guanine, and uracil. Adenosine triphosphate, otherwise known as ATP, is a multifunctional nucleotide known as the molecular currency of intracellular energy transfer. Like tiny rechargeable batteries, ATP molecules transport chemical energy within a biological cell. These molecules can move energy around because the phosphate groups contain a lot of potential energy, which is released when these bonds are broken. Our final ingredient we'll be using in our macromalad is the salad dressing. As you can see, there is separated oil in this bottle. Those are lipids. A lipid is a fatty organic compound that is insoluble in polar solvents, such as water, but soluble in nonpolar solvents, such as ether. Its major functions are for energy storage, as a structural component of the cell membrane, and for cell signaling. It is usually made up of glycerol or fatty acid units, with or without other molecules. Fatty acids are the monomers of lipids. Many lipids are amphiphilic, meaning they have both hydrophobic and hydrophilic components. An ester linkage occurs when a carboxylic acid reacts with an alcohol group, producing a water molecule. A triglyceride consists of a glycerol and 3 fatty acids. Ester linkages are what create the bonds between the fatty acids and the glycerol. Triglycerides are the main component of body fat in humans and other animals, as well as vegetable fat. Fats can be both saturated or unsaturated. Saturated fats are solid at room temperature, come from animal fats, and have no double bonds in their chemical or molecular structure. Unsaturated fats are liquid at room temperature, contain double bonds in their molecular structure, and usually come from vegetable matter. This salad dressing is easily identifiable as an unsaturated fat. As you can see, it's liquid at room temperature. And voila! The macromalad is complete. Enjoy!